Hello all, welcome back to the alumni series talk series brought to you by the Biotech Research Club under the umbrella of Biotech Student Association. Today we have with us Dr. Aman Kumar. Aman graduated from the Department of Biotechnology at IIT Madras with a dual degree in biotechnology in 2015. He was also a core team member of the IGEM 2013 batch while at IITM. Uh, he went on to do a PhD at the University of Texas Southwestern Medical Center, where he worked on host pathogen commensal interactions in the human gut in the lab of Professor Vanessa Sperandio. He is currently a postdoctoral research associate in the lab of Professor Andrew Goodman at the Yale School of Medicine. Today, he'll be talking to us about the interplay between tryptophan metabolites and the virulence factors of enteric pathogens. We look forward to having you join us for the talk. I request you all to keep yourselves on mute and turn off your video cameras during the lectures, unless you have a doubt. Following the lecture, we'll also have an informal discussion on academic trivia such as research internships, grad school applications, and so on. Over to you, Aman.
Uh, yes, thank you for that introduction. Can you see my screen?
Yes, yes.
Okay. Uh, Again, thank you for the wonderful introduction. I'm very happy to be here. And today I'm going to talk about my PhD work that I conducted in the lab of Vanessa Sperandio at UT Southwestern Medical Center. And I'm currently a postdoc in Dr. Andrew Goodman lab at Yale University. So what you're seeing in the background here is one of the buildings of UT Southwestern where I've conducted my research. And I like to have this picture in my introductory slide just to give a sense of where it actually happened. So before I go into the talk, I want to briefly acknowledge people who have helped me throughout this journey. Uh, my research interest actually sparked when I joined IGEM team. And I was a member of IGEM 2013 team. And these are all the folks who have helped me throughout the journey. After that, I did my undergraduate research with Dr. Nitish Mahapatra, and they were all like wonderful people. I had a great time here. Following which, I went to do my PhD in the Sperandio lab at UT Southwestern Medical Center, and then currently I am a postdoc at Yale University. And of course, there are many, many more people who have helped me throughout this journey, and I'm thankful to all of them. So let's just jump in. One of the questions that actually was very interesting to me was that how pathogens sense gut info chemicals and regulate expression of its virulence genes. So, before I go into the talk, if you have any questions and if you want to ask, just feel free to unmute yourself and ask. Or if the voice is cutting or something, just let me know because I can't see any of your faces here. Okay, so as we all know that our GI tract contains trillions of bacteria. And because there is trillions of bacteria, there are trillions different types of metabolites that are being produced either by bacteria. They also come from diet. They also come because we take so many medical drugs. And also host can produce different types of small molecules. And therefore, our gut is very densely packed environment, which is filled with different types of small molecules. So this is just a cross-sectional view of a colon. And what you're seeing here is the luminal environment, which is, so when you look at the colon, there is it's not a hollow tube, it's a densely packed environment. And this luminal environment actually, uh, let me just choose my pointer. Yeah, this luminal environment actually contains trillions of bacteria. And this trillions of bacteria can produce different types of small molecules. And when we zoom in a little bit more, there is spatial heterogeneity. And what we see is that different types of small molecules are being produced by the host. As you can see here, that host gut epithelial cells can produce different types of small molecules. And many other small molecules are also produced by the bacteria present in the luminal environment. So the question is that when a pathogen infects you, it encounters an environment which is densely packed and also filled with different types of small molecules. So how does this pathogen sense these small molecules and find a niche that is more conducive for its growth? For example, this particular pathogen has to come close to the epithelial surface where it can infect the cells and cause disease. Towards that end, I started working with tryptophan and its metabolites. As we all know, tryptophan is an essential amino acid and has to be taken from our diet. Tryptophan can be directly metabolized by bacteria using bacterial metabolism to indole. Gut microbiome can convert tryptophan to indole. Host can also convert tryptophan. One of the ways by which host can metabolize tryptophan is through the serotonin pathway. Tryptophan can convert tryptophan can be converted directly to serotonin by host. These cells, which are enterocomorphin cells, they are present in the gut and they can convert tryptophan to serotonin and release in the luminal environment. So the overarching aim of my PhD thesis was to study the role of these bacterial and host-derived tryptophan metabolites during bacterial infections. And I'm going to talk about two of my projects. The first project was related to indole, which is a bacterial derived small molecule, and how it signals in host pathogen microbiome interactions. And the second project would be related to serotonin coming from the host side and how it regulates virulence of enteric pathogens. Okay, for the first project, let me give you a brief introduction on indole. Indole is a small molecule. It's long been known as a signaling molecule, and it is implicated in biofilm formation, virulence factor production. It is known to be present between a concentration of 250 to 1,000 micromolar in human feces. And in the gut, indole is produced using this tryptophanase enzyme. So bacteria can convert tryptophan to indole using tryptophanase enzyme. It is also known to freely diffuse in and out of the bacterial cell via passive diffusion. And a few years back, it was shown that 1 in 6 microbial species in the gut has tryptophanase enzyme and can produce indole. What it means that indole is quite abundant in the gut. And therefore, the question is that what is it doing in terms of host microbiome pathogen interaction, right? This is an abundant molecule. And if there is a role it plays in this interaction, and that is what I got interested in. So the pathogen of choice in the lab was AE pathogen or attaching and effaging pathogen. So I was super impressed with this design. So I had just put this flyer here where you see that this particular pathogen is trying to invade the cell. Although there is one detail that was missing. The pathogen that I work with is not an invasive pathogen. It's an attaching and effacing pathogen. So what it does is that it attaches itself to the host cell. It does not go inside the cell. And these AE pathogens are gram-negative pathogen. They are transmitted via contaminated food and water. One of the pathogen from this family is EHEC, 0157S7. I don't know if some of you have gone through our iGEM project in 2013. We also proposed something with EHEC, although there is no correlation between my iGEM project and this. This was just a coincidence that I happened to work with EHEC. So this human pathogen is responsible for multiple outbreaks of bloody diarrhea and HUS worldwide. Now, this is a human pathogen, and the problem with EHEC is that in order to study this host pathogen interaction in vivo, we make use of mice, but EHEC cannot infect mice. So we use a surrogate for EHEC, which is Citrobacter rodentium. It's a mouse pathogen, and it's a widely used surrogate model for EHEC. So for the rest of my talk, I'll be talking about when I talk about a pathogen, it is reflecting C. rodentium, which is a mice pathogen, which is very much similar to EHEC. So now what makes EHEC pathogenic or C. rodentium pathogenic is because of this pathogenicity island, which consists of multiple operons, different clusters of genes. And because of the presence of this pathogenicity island called locus of enterocyte effacement or LEE, EHEC can form a type 3 secretion system. Type 3 secretion system is basically a needle and syringe kind of structure, as you can see here. And this is from the bacterial side, where there is inner membrane of bacteria, outer membrane of bacteria. Now, because of the presence of this type 3 secretion system, bacterial effectors can translocate from the bacterial side to the host side. One of the translocated effector is tear, which then binds to intermin present on the outer membrane of bacteria. Now, this completes the process of attachment of bacteria to the host cell, and bacteria can now start injecting its effectors or virulence factors into the host cell and cause disease. If you try to look it under the microscope, it will look like this, where this bacteria is attaching itself to the host cell, where in a process of which looks like a pedestal formation, right? And this happens because there is a severe actin rearrangement that takes place beneath the bacterial cell, and it appears like a pedestal structure. And at this point, I want to also emphasize that if you have a bacteria or a pathogen which lacks this type 3 secretion system, this pathogen becomes avirulent. So it can no longer cause disease. And therefore, in the lab, most of our strategies are to target this type 3 secretion system. Uh, so if we decrease the expression of this type 3 secretion system related proteins or genes, we are also decreasing the virulence of this pathogen. Okay. So the question that I wanted to address was, does indole, which is an abundant molecule, small molecule in the gut, play any role in modulating the virulence of this particular bacterial pathogen in the gut? And to do that, we wanted to first understand how is indole distributed in the gut, right? So again, I'm showing you the cross-sectional view of the colon, which is inhabited by trillions of bacteria, collectively known as the microbiome. And if we zoom in a little bit, we see that this trillions of bacteria can produce indole. As I said, that indole is a bacterial derived molecule. So it is being produced by the bacteria. So most of this bacteria actually resides in the anaerobic part of the lumen. So as you go towards the epithelial surface, it becomes more oxygenated or aerobic, and there is more anaerobic. So you have like more dense population at the middle center of this luminal environment. And so we hypothesize that there would be higher concentration of indole here, and there will be lower concentration of indole near to the epithelial surface. We measured that by mass spec, and we find that indeed the concentration of indole in the luminal environment was higher as compared to indole in the near to the tissue or in the tissue. We also measured the concentration of a precursor molecule, tryptophan, which was constant in these two environments, suggesting that indeed in the gut there is a gradient of indole present. So because there is a gradient, this pathogen is also encountering different concentrations of indole. So when a pathogen infects you, it's probably in this environment where there is a lot of indole. Now it has, in order to make an infection, it has to move from this environment and come closer to the epithelial surface where it can attach itself and cause disease. And there is low concentration of indole. So I wanted to understand how does a pathogen responding to different indole concentrations in the gut. And to do that, we did some mice experiments, and we started manipulating the concentration of indole in the gut. And for the first strategy, I employed B theta. B theta is uh, or Bacteroidetes theta iota micron. It's a prominent member of the microbiome. And B theta also harbors this TNA gene, which makes tryptophanase, and it can produce indole from tryptophan. And for the second strategy, I engineered C. rodentium to produce indole. Now, C. rodentium by itself, C. rodentium is a pathogen again, is by itself, it does not produce indole. So we have to engineer C. rodentium to produce indole. So this is a schematic of the experiment where we had in one group, these group of mice were like both the groups of mice were microbiome depleted. And then they were inoculated with wild type B theta that can produce indole as indicated in purple color here. And then we infected these mice with Citrobacter rodentium, which is in red. For simplicity, the microbiome is represented in green and pathogen is represented in red. Uh, in the second group, we inoculated mice with a mutant of B theta that cannot produce indole, which is a TNA, delta TNA B theta. And then we infected these mice with Citrobacter rodentium. We also measured the concentration of indole recovered from the feces of these mice. We find that mice that were given wild type B theta had abundant levels of indole, while the other group did not. So when we did the experiment with mice where we first depleted the microbiome, we inoculated these mice with either wild type or a mutant of B theta that cannot produce indole, infected these mice with Citrobacter, and we collected stool samples. We find that the expression of virulence genes that are responsible for formation of the type 3 secretion system was upregulated in the mice that were given a mutant of B theta that cannot produce indole. It means that the presence of indole was able to decrease the expression of the virulence genes, suggesting that B theta can limit serodentium infection. And for most of my presentation, I'll be just showing key data and not like a lot of data. Okay, so for a second strategy, we engineered serodentium to produce indole. Now, indole can be, so E. coli produces indole, and it has this operon called TNA-CAB operon, which is responsible for production of indole. We took this operon and engineered into serodentium, and now serodentium can produce indole. Again, we made use of microbiome depleted mice. We infected these mice with indole producing serodentium and the other group of mice with indole negative serodentium. When we measured the concentration of indole, we find that mice that were infected with indole producing serodentium had abundant levels of indole, while the other group did not. And again, when we did this experiment, we had an amazing phenotype where we see that mice that were infected with serodentium with indole producing ability had much higher survival rate, and most of the mice ended up surviving the infection, while the other group that were given wild type serodentium had higher mortality. This also correlated with decrease in the virulence gene expression. So this experiment also suggested that yes, indeed, indole can decrease serodentium infectivity. So for part one, what I've shown you is that the presence of indole in mice system or mice gut was able to limit the virulence of a pathogen, rodentium, and it also led to prolonged mice survival. So I'm going to switch gears and talk about my second part of my project. So in the first part, I talked about bacterial metabolism, which led to production of indole. But host can also metabolize tryptophan. One of the pathway through which host metabolize tryptophan is through the serotonin pathway. Serotonin is produced from enterochromafin cells in the gut. And why it is interesting to study serotonin is because 90% of serotonin is actually produced from enterochromafin cells present in the gut. And it has been observed that serotonin, when you have altered serotonin signaling, it led to various gut disorders, including IBD. And it also plays an important role in immune cell function during inflammation. And it has been known to regulate gut motility and maintains host metabolic homeostasis. So what you see is that we know a lot about serotonin as a neurotransmitter in the brain. And we also have a lot of information on serotonin from the host perspective. But we know very little about what it can do to a pathogen. And that's where I wanted to understand if serotonin, which is being produced in such huge quantity in the gut, can also affect pathogens' ability to respond. So the question was, does serotonin modulate virulence of these bacterial pathogens in the gut? And to
Aman, sorry,
understand that,
can
yes,
I interrupt? Yeah,
yes, sure.
I didn't understand in the initial part how exactly did the gradient play a role, or was only the presence and absence of indole analyzed?
Yes, I have not shown you that information, but okay, let me just go back a little bit. Yes. So gradient played a role because you have, yeah, presence and absence is important. But what I have not shown you, and probably it will come in the later part, where, as I said, that indole can be diffused into bacterial cell, can passively diffuse. And therefore, when you have higher concentration of indole, the likelihood of indole to diffuse into bacteria and be in the periplasm is higher. That's where the effect takes place. Uh, does it
Yeah,
answer you?
okay.
Yeah, so higher
Yes, yes.
concentration allows it to diffuse. If you have low concentration, it cannot diffuse, right?
Yeah,
And yeah, so
true.
that comes into the mechanism part, and I'll be talking a little bit about that. And probably that will clear later on.
Yes, thanks.
That's a great question, though. Yeah. Okay. So the fate of serotonin in the gut. So what happens is that tryptophan is converted to serotonin using this pathway, where tryptophan is first converted to 5-hydroxytryptophan using this enzyme called tryptophan hydroxylase, which is further decarboxylated to serotonin. And once serotonin is formed, it is released into the lumen, and then it can again be reuptaken by this particular transporter called SERT, or serotonin reuptake transporter. And this is how the homeostatic level of serotonin is maintained. But at the same time, we also get strategies to start manipulating the concentration of serotonin. I also want to highlight that this particular enzyme is rate limiting enzyme of this pathway. So we use three strategies to start manipulating luminal serotonin levels in the mice gut. Uh, for the first strategy, we employed SERT knockout, which will not allow serotonin to be reuptaken back into the cell to change the concentration of serotonin in the gut. For the second strategy, we made use of a pharmacological inhibitor of this rate limiting enzyme TPH1. And for the third strategy, we used an inhibitor, or which is a selective serotonin reuptake inhibitor, to change the concentration of serotonin. So let me go one by one through all these strategies. For the first strategy, the SERT knockout mice. As I said that SERT, which is a selective reuptake transporter, can transport back all the serotonin which is being released into the lumen back to the cell. Now, when you have a knockout, you can imagine that there will be higher concentration of serotonin present in the luminal environment. We also measured this concentration between the wild type and SERT knockout mice. We find that there was higher concentration of serotonin present in the luminal environment as compared to the wild type mice. Uh, so in this case, we again performed this experiment using either the wild type mice or a SERT knockout mice. We infected both these group of mice with Citrobacter rodentium, and we collected stool samples and monitor for survival. In terms of survival, we find that the SERT knockout mice infected with Citrobacter rodentium had much lower mortality rate, and 50% of the mice ended up surviving. This also correlated with decreased pathogen burden, where we see that the SERT knockout mice had lower pathogen load as compared to wild type mice, suggesting that accumulation of serotonin in the gut was able to decrease Citrobacter rodentium infectivity. For the second strategy, we made use of this PCPA, which is an inhibitor to the rate limiting enzyme of serotonin biosynthesis pathway. Again, using this PCPA, we can imagine that there would be a lower production of serotonin. And that's what we find, that addition of PCPA leads to decrease in the serotonin levels in the gut. And when we perform this experiment, we find that in terms of survival, mice that were given PCPA had much lower survival and faster death as compared to mice that were given just PBS. This correlated with increased virulence gene expression of bacteria recovered from mice that were given PCPA, or mice that had lower concentration of serotonin. This again suggested that when you decrease the concentration of serotonin in the gut, it will lead to increase in C. rodentium virulence. So as a final pass, we wanted to just confirm our phenotype by using a widely used antidepressant, which is Prozac. I don't have data about India, but in the US, 1 in 10 people in the US actually use Prozac as an antidepressant. So when you have Prozac, what you can imagine, it inhibits this SERT, the reuptake transporter, and therefore you can think that there will be higher concentration of serotonin available, similar to the SERT knockout phenotype, right? And when we did this experiment, what we find was that mice that were given Prozac had lower pathogen burden as compared to mice that were just given water. And in the second group, mice that were given, it is also correlated with the expression of virulence-related genes, where we see that mice that were given Prozac had lower expression of virulence genes of Citrobacter as compared to mice that were given water. Again, so to summarize this part of my story, what I've shown you today is that if you decrease the luminal serotonin levels here in the gut using different types of pharmacological inhibitors such as PCPA, this leads to increased virulence of bacteria and also increased pathogen load. While when you try and increase the serotonin levels in the gut by using a SERT knockout mice or Prozac, this will lead to decreased virulence and also decreased pathogen load. So at this point, I have shown you all the phenotype and everything, but you might be wondering that how it is happening. What's the mechanism, right? And that is what we were all thinking, that how is bacteria sensing this indole and serotonin? So what we did was we performed an RNA-seq experiment where we used pathogen with and without indole uh, or serotonin. We extracted the RNA and we did RNA-seq, filtered differentially expressed genes. And at this point, we had this hypothesis that, and also like information in literature, that small molecule signals can be sensed by bacterial membrane-bound histidine sensor kinases. So in bacteria, the signal transduction happens through this sensor kinase, usually through the sensor kinases, which forms a part of a two-component system. It has a sensor kinase and then a response regulator. These sensor kinases are located in the inner membrane of bacteria in this pathogen. It's located in the inner membrane. And when we did screen for all the histidine sensor kinases, we find CpxA as the candidate indole and serotonin sensor. Now, again, I'm not going to go into detail, but I'm just going to give the model and a key experiment for this particular identification of this sensor. So as the previous question, what what we thought that when you have a higher concentration of indole, indole will diffuse into the periplasmic space. So you have inner membrane. I'm not showing the outer membrane. So indole can diffuse, indole or serotonin can diffuse into the inner membrane, where it can be sensed by this membrane-bound sensor kinase CpxA. Sensing of this signal by CpxA leads to its decreased activity, which causes decreased activity of its response regulator, which is just beneath this sensor kinase. And now this can no longer bind to its DNA targets and lead to decrease in the virulence gene expression. And that is our model. And to understand, I'm just going to show you the key experiment. What we did was we made use of liposomes, and we loaded this liposome with CpxA, purified CpxA, in the presence of signal, which is either indole or serotonin. And we also added tryptophan as a control. And we added radio labeled ATP. And what we find is that with time, because this is a sensor kinase, it should phosphorylate itself. That is the normal function of a kinase. So with time, there is increase in the phosphorylation activity of CpxA. But when we added this signal, which is indole, it inhibits its phosphorylation activity and leads to decrease in the phosphorylation activity of CpxA. However, when we did the same thing with tryptophan, we don't see that effect, suggesting that indole is being sensed by CpxA. Similarly, we did that experiment with serotonin, where we find that addition of serotonin lead to decrease in the autophosphorylation activity of CpxA. So this experiment suggested that indole and serotonin is functioning through the CpxA and CpxR node. So for the last few minutes, I'm just going to talk about the model. So the model of this, the model that we finally came up with was when you have bacteria present in the zone of high indole or serotonin concentration, what happens is that indole, which can diffuse also, this high concentration enables indole to diffuse into the periplasmic space, where it is sensed by this membrane-bound histidine kinase, CpxA, which leads to its decreased activity and therefore decrease in the virulence-related genes. But once this particular pathogen find a niche that has less concentration of both indole and serotonin, what it does is that CpxA remains in its active state. So it remains in the phosphorylated state. It can phosphorylate its targets like CpxR, and which leads to increase in the activity of virulence genes. So essentially what we found was that having this ability to control, to judge its space, a pathogen is able to understand where to express its virulence genes. And when it finds a space or niche that is more suitable for its expansion, it goes there, attaches itself, and causes disease. And with that, I would like to acknowledge people in the lab, because all this work was conducted at UT Southwestern. So my PI, who is Vanessa Sprandio, all the lab members, my thesis committee, William's lab, who helped me with the mass spec experiment. All the images were done with BioRender, and my funding source. And I think I'm on time, so I can take any questions if you may have at this point.